Now, if we don't find anything, if there is possibacillary state, bacteria, maybe there are 10, 15, 100 bacteria in per ml of sputum, then what to do? I am focusing on pulmonary tuberculosis just only because only then only you will be able to compare it with extrapulmonary tuberculosis. Pulmonary tuberculosis means only lungs. Even there is pleural effusion, it is called extrapulmonary. So for culture method, we have this is the gold standard of diagnosis. Previously, we used to do it on slant LJ media with malachite green. So on a green base, there used to appear buff colonies, but it took almost 3 months, 4 months for the colonies to develop. And there were biochemical methods of diagnosing what bacteria it is. May it be nitrate reductase test or aryl sulfatase test or rapid Bohr test, iron uptake test, whatever. But now I think they are obsolete. So nobody is doing it. Now this thing was, this method was used for some time, few years back. It is liquid culture media, liquid bath was used, and it was a radiometric method. So radiometric method used to be harmful for lab technicians. So a Bactec 960 method, which is also known as MJIT method, that was developed because in MJIT method, in MJIT method we used to add penta, 5 antibiotics in it to reduce contamination, to reduce cross infection with the bacteria. Penta was, I think it was polymyxin B, amphotericin B, nalidixic acid, trimethoprim, and some azadirachtin or something. So 5 antibiotics were there. So both the things were decontamination and growth was done in the same tube. So there were chances of next contamination were very there, and all the reports were available within 2 months, less than 2 months, maybe 45 days. Then in direct evidence, they were all, both of them, the culture techniques, whatever on solid or liquid, they are phenotypic methods. Because seeing the bacteria, they form of the bacteria. But there is another method, this is genotypic method. Then we are diagnosing, we are seeing the DNA of the bacteria. With the help of DNA, we are diagnosing. So we can be doubly sure that this is Mycobacterium tuberculosis. So there are 3 ways of doing this thing. They are all PCR based. PCR, all of you must be knowing that the DNA is actually heated, it is separated, so it is called denaturation. Then it is primers are already added in it. Dr. Mani, primers are already added in it. Added in the cartridge.
अच्छा इसकी बात हाँ सर
And they
closed system
add 2 primers are there. 2 primers with the codons and everything all in one cartridge, and DNA separates. Then there is these primers add to it, and the template is formed, and then DNA polymerase starts its action. This is not the polymerase which our body has, because it is heat labile polymerase. This is TAC polymerase, temperature resistant polymerase, because in thermocycler we heat it to 90-95 degrees, then we cool it, then we again raise the temperature. So our polymerase will not be able to tolerate this.
So which one is superior, CVNet or TrueNet?
TrueNet is better. So CVNet cartridge based, commercially it is also known as Gene Expert. So it is also known as Gene Expert MTB/R, reform casing, RIF. because if you add a third primer with RPO gene, it will tell you whether there is rifampicin resistance or not. Simultaneously, you can detect it is mycobacterium plus it is resistant to rifampicin or not. This is all sensitivity, specificity. A TrueNet is a small machine, which is almost the size of a laptop. It is being used in periphery also, in India.
I think same machine is used for COVID test also, na? Same machine.
Yes, same machine can be used for RT-PCR also. It is also based on PCR. These all the CBNet, TrueNet or LPA, line probe assay, they are all PCR based. Initially the PCR is done, the duplicate copies of DNA are made, and then they are made. So here again the results are available in 1 and 2 hours, and TrueNet, MTB, these 3 types are available, but we are using TrueNet, MTB plus for detection of MTB and this thing. Other are for resistant cases. The beauty is that it is an isothermal machine. In CBNet we were using